It's my pleasure to introduce Markus Diesmann. Markus holds a PhD in physics from the University of Bochum. Then he actually conducted that PhD work at the Diesmann Institute of Science and also at the University of Freiburg. In 1999, he joined the Max Planck Institute for Dynamics and Self-Organization in Göttingen. And then from 2004 to 2006 was appointed as a junior professor for computational neurophysics at the University of Freiburg. Then between 2006 and 2011, Markus was leading a team at the Riken Brain Science Institute in Japan. And then in 2011 is now director at the Institute of Neuroscience and Medicine, also at the Computational and Systems Neuroscience Institute and the Institute for Advanced Simulation and Theoretical Neuroscience at the Jülich Research Center, right? As some people in this room know, he's also kind of co-headed the NEST tool, which is a pretty impressive computational neuroscience simulation tool. And so, without further ado, also I'm pretty interested about sequence learning, so please, the floor is yours.
Thanks. Thank you very much for this kind introduction, even a double introduction. I never had this. And also thanks for the invitation. I know that this is a diverse audience, so I will try to do my best to explain something to everybody. If there are then some boring sections, I hope that there's at least one for whom this is of interest. On this one. Well, I don't know. Maybe I need to be much more. So let me start like this. If we look at modern AI, then it excels on many tasks. But brains are still unbeaten on many natural tasks. For example, learning from two examples, or the high-end coordination that robotics has to do. So I think there's still motivation to understand more about how the brain works. But another aspect is that the AI algorithms that we have today, they are optimized for present-day computers, and because of this, we pay a terrible price, and this is this discrepancy that the brain uses about 20 watts, whereas a present-day supercomputer uses about 2 megawatts, and actually the next generation supercomputer that is going to be built at our site will use 20 megawatts. So there's really no progress here. And at the same time, we see the end of Moore's law, so the number of transistors per surface area chip is no longer doubling. So we can also not expect that this problem is be removed by just miniaturization. So we need completely new principles to overcome this, and maybe these principles we can extract from the brain. And then you can ask the question, is there any hope that we can achieve this? And this hope is here expressed on the right side. Here you see a photograph of a nerve cell in the brain. And from the length scale here, you see that the diameter of the cell body here is about 20 microns. And the photo here shows a modern transistor. So you see this typical 3 pins. And here the length scale is 20 nanometers, meaning that in our technology today, we can already produce structures that are 3 orders of magnitude smaller than the relevant structures in the brain. And a bit of a different view on this. When you move the slide forward, is expressed here. If you look at the number of neurons in the cortex, then we have about 10 to the power of 10, but this is not more than the number of transistors that we have in a modern processor. So also the number of elements does not seem to be the problem. Next slide, please. So what is then the problem? The problem is the wiring. On the right side here, we see again a photograph. The length scale is 20 microns again, but what is colored here in black are now the outgoing structures, the axons of the nerve cells. The white parts are not nerve cells, but blood vessels. And you see that this volume here is completely densely covered with cables. There are about 100,000 neurons in each cubic millimeter of brain, and each neuron has about 10,000 contacts with other neurons. And this results then in 3 kilometers of outgoing cable within this cubic millimeter. And this is a density of packing that no technology that we have today can achieve. But maybe we don't have to achieve this, because as I said, the structures that we can produce are 3 orders of magnitude smaller. And because of this, also the switching times of electronics are much higher than of biology. So we still have 3 orders of magnitude room for playing with multiplexing and other technologies. So the whole thing is not completely hopeless. Next slide, please. Can you go back once? Sorry. But is there hope that we can understand the function of the brain? And in order to motivate this, I have brought you this slide here. These are cross sections through the brain of mammals, from mouse to human. And what you should see here is that in the course of evolution, the basic structure of the cortex has not changed much. But what has changed is the volume of the cortex. It has expanded by 3 orders of magnitude. So apparently, nature hit on some principle that it was so successful that it was worth to maintain it and to just build more of it. Next slide, please. And there is another universality that we can see. And this is that also over different sensory modalities, the structure of this part of the brain is very much conserved. So although something completely different is processed, the brain looks more or less the same, so that we can assume what actually then determines what is being processed is more the way how these different parts of the cortex are connected than the changes within the local microcircuit itself. So it's this dual universality that gives us hope that there is something to be discovered. Next slide, please. And over the years, theoreticians have worked on understanding this local computation that is going on in the cortex. I've brought here a number of studies. We have also contributed one to this list that I will show you in a bit more detail. And what is happening in the past few years is that now, due to the progress in technology, we can model and simulate these networks at the full scale. So with all neurons and all synapses, meaning that we can remove any doubts on scaling properties and so on from these models. Next slide, please. And just another funny reminder on the severity of the problem. This is a slide that I received from Shahar Kvandinsky, a theoretician at Haifa University. You know that a computer can now win against a human, probably the most complicated game that we have, game of Go. But you also have to consider the price of this. And the electricity bill alone is $3,000 per game, and this is not considering the hours of training that went into this. Next slide, please. And the basic interactions that I will use today in the models that I'm describing is this one here. This is the integrated fire model, and therefore I will describe it to you in a bit more detail. So what we assume that each nerve cell maintains a potential between the inside and the outside of the cell. So if you would inject a current into the cell, then one can observe how the voltage is changing over time. And you see that when the current is switched on, the voltage increases, stabilizes, and when the current is switched off, it falls back to a resting level. Only when the current is big enough, that eventually a threshold is reached, and then a sharp pulse is emitted. This is called an action potential, and only these sharp pulses are then transmitted to the next neurons, and in the receiving neurons also a small voltage excursion is elicited, and this small voltage excursion is called a postsynaptic potential that occurs with some delay of a few milliseconds in the postsynaptic cell. And by comparing the voltage scales here, you already see that many of these postsynaptic potentials need to overlap to cause an action potential in this postsynaptic cell. Now, this picture here is very much what an experimenter sees if such an experiment is carried out in a dish where the neuron is basically isolated from the rest of the other neurons. It really looks very similar. However, if you would record this memory potential here in the intact brain, in a living cell, then these traces look very different. This is depicted here. Here's again voltage over some stretch of time. And you see that there are huge fluctuations of the memory potential that basically fill all the space between the resting level and the threshold. And at seemingly random times, their action potentials are emitted at a low rate of a few hertz. What we know now today, that these fluctuations here are not noise. The neurons are basically operating like deterministic machines on the voltage scales that are of interest for us here. These fluctuations are coming from the superposition of such postsynaptic potentials that the neurons receive at any moment in time over 10,000 connections to other neurons. 80% of the neurons in the brain are of this type, that they cause these so-called excitatory postsynaptic potentials that drive the membrane potential towards threshold. 20% are of the opposite type, they drive the membrane potential away from threshold. So this is everything you need to know about the basic dynamics of neurons here. Next slide, please. And so based on this, a few years ago, we decided to build a model of a cubic millimeter of cortex, where we decided that the neurons all have identical properties, and even the inhibitory neurons have the same properties as the excitatory ones. The only thing that distinguishes them is that inhibitory neurons make this inhibitory postsynaptic potentials. And we made this simplification because we wanted to find out how the structure of the network influences its dynamics. And so in compiling these connection probabilities between the different neurons in the different layers that the cortex shows from the surface of the brain into the depths. So I'm trying to continue my talk. I hope you'll remember what I have said. So on the right side here, we see the depiction of the activity in this circuit. The spikes are marked by dots. And in order to guide your I have sorted the spikes according to whether they come from excitatory neurons, these are marked in blue, or from inhibitory neurons, these are marked in red. Of course, in the brain, this is in reality, this is all intermixed. But you can immediately make some observations here. First of all, the spike rates are different in the different layers, as they should be. And the activity is asynchronous, irregular as it should be. And also, the spike rates of the inhibitory neurons are systematically higher than the ones of the excitatory neurons. And this is also according to the experimental data, and it's not something that we built in. I told you that we decided to make the dynamics of the excitatory and inhibitory neurons really identical. This is all coming out of the structure of the network. We published the formal description of the model at some website. And next slide, please. And the success of this was that this model was picked up by other laboratories that were doing more interesting experiments with this, or were extending the model. This is something that we wanted to achieve. Next slide, please. But of course, one can also criticize this model because it is describing all the synapses that are within this cubic millimeter, but each individual neuron receives only 50% of the synapses from the local circuit and 50% from outside sources. So you could as well say that this circuit is largely underdetermined. The structure that the brain exhibits, if you zoom out a bit more, is that the neurons are organized into brain areas where the probability of connection decreases with the distance of the neurons. So there is a spatial organization on the level of brain areas. And then if you move out even further, you find out that in the human, for example, the brain is organized into about 100 areas, but each area is only connected to a few of them. So we have to talk about a network of networks with at least 3 levels of organization, and even on the highest level, there is still room for an interesting recurrent network. Next slide, please. So we set out to capture more of this circuit, and we used as an example the visual system of the macaque monkey. Not because we were so much interested in the macaque, but just at the time there was a very good anatomical database available. We decided to model the 32 areas and find out about how this long-range connectivity is interacting with the local connectivity. And in order to keep the total number of neurons under control, we placed just one of these microcircuits in each of the areas. And also here, by simulating such a circuit, we could make some interesting observations. For example, that we could reproduce the functional connectivity as you would obtain it from fMRI studies. Next slide, please. But what this way of modeling, which you can term bottom-up modeling, does not give us is insight about network function. So we started to combine this with work on what these circuits can actually do. And I can report to you how far we got here. We decided to focus on the specific problem of sequence processing. Sequences occur naturally in many tasks that the brain has to do in the processing of sensory inputs, but also in the planning of motor activity, language comprehension, and production. A famous example of such sequences is how we move our eyes over an image, for example. So that brings us to the hypothesis that maybe sequence processing is a generic type of computation that the brain has to do, obviously not the only one. So what would be the task of sequence processing? Would be the prediction of upcoming events in a context-dependent manner, the detection of non-anticipated events, autonomous replay in order to produce something, the segmentation, the naming or classification of sequences, and also the merging of sequences into higher ones. So how can we operationalize part of this? This is depicted here. If you think about input sequences ADVE or FDVC, then the task of the brain could be that once A was presented, a prediction would be made that now D should come, which would then be confirmed or not confirmed, and so on. And obviously, if you start from scratch, from a network that has not learned anything, then there could be no prediction, and a mismatch would be detected. However, after training, we should have a situation where if A is presented, that D is predicted, and when then D really comes, there's no mismatch, and then in this case, B should be the next predicted value, and then if B comes, everything is fine. And the same should happen if the alternative sequence FDVC is presented, but then if ADB is presented, and then as a last element, C comes, which is the end of the other sequence, then that would be a mismatch. So that's the basic computation that we want to study further here. Next slide. And here we are starting from a model that was presented by Jeff Hawkins and Ahmad in 2016. It's called the Hierarchical Temporal Memory model, but we will only consider a small part of this. By the way, Jeff Hawkins describes his ideas in this nice book here on intelligence, and he has also written a further book. I find this book very inspiring and recommend it to you. So they put forward a model that is solving this sequence processing as I described it. However, they do this in a very abstract dynamics that uses discrete time or no dynamics at all, if you want to call it this way. They require that neurons are ternary, so that they have three states: quiescent, predictive, and active, and they employ a non-biological form of plasticity. So they have both of these components. They are thinking about what the local microcircuit can do. They distinguish the distal basal dendrite in its computational role for processing context, and they consider another pathway for feed-forward information. But on the point of dynamics, it's not really clear how this can be biologically realized. So we set out answering the question whether it's possible to find a biologically plausible implementation of this that is based on spiking neurons, as I described them to you before, and is not violating any of the biological constraints that we know today. And we have described our results in this paper here that you can read up. Next slide, please. And the solution we found is the following. Our spiking model is characterized by continuous. It's a continuous time spiking version of the TM model. It shows sparsely randomly connected networks of excitatory and inhibitory neurons. excitatory neurons are stimulated according to stimulus preference. I will show this to you. And we use nonlinear integration of synaptic inputs as they occur in the brain. And the whole thing is guided by an unsupervised learning of sequences by a homeostatic form of plasticity. So here you see a sketch of this model. We are now concentrating only on this these two sequences that I introduced to you at ADBE, FBE, and for each of these symbols, we have something like a receptive field, if you want to call it this way, like a population of neurons that is coding for this stimulus. If A is presented, or the neurons of A will fire. And this is these are excitatory neurons, and they are recursively connected to a pool of inhibitory neurons. And in the beginning, the activity in this network is just random, but the conditions in the cortex, in the local microcircuit, are such that basically every axon is touching every dendrite. So any pair of neurons has a chance to form a synapse. At any point in time, only 10% of this is realized, but in principle, a synapse can be grown everywhere. And the connectivity that is resulting after learning, you see here, and I will guide you through this a bit. So if we start, what would happen if A is presented, then these are the source neurons, and the deactivated synapses are marked here then in this light blue, and you see that there are then synapses developing to the population D. So this would be this pathway here, but you see that not all the synapses are activated here, and this is because there is a winner-take-all mechanism going on. When A is activated, then all the neurons of A are activated, but in this learning process, only a certain fraction of the D neurons fire, and therefore only a certain fraction of the synapses are strengthened and established.
What is the x-axis within one of these squares?
This is just number of neurons within this population. So it shows the connectivity matrix. Yeah. Now, if we consider the other sequence, F, then we see that there are also a set of synapses from F to D, but these are different synapses. And now comes an interesting point. Now we are in D, and we see the connections between the symbol D here and the symbol B, which is the next one, in both of these sequences. And we now see that we have both colors, shades of blue here. And that means that activity is flowing from D to B across different synapses, depending on what the context is. Whether in the station before, this was coming from A or from F. Yeah, and this is why in this graph here you see also the connections by two different shades of blue here. So this is the connectivity that is developing by this mechanism, and what is now happening in the activation of such circuit is shown here. We again have our populations here, and now we assume that A is presented, then the neurons in the population A are firing, and they are all firing because they are all stimulated. And in response to this, the neurons that are connected by synapses from A in the population D get this spiking input, but the dynamics of the neurons is such that in the distal basal dendrites, where these synapses are, the so-called dendritic action potential, or NMDA spike, is elicited. So this is not enough to drive the neuron, the postsynaptic neuron, to emit a spike on its own, but it's an activation in the dendrite that is long-lasting. And this is depicted here in this graph. So upon the presentation of A, the neurons that are connected in D have this long-lasting NMDA activation. And if then in the next step of the sequence, D is stimulated, then this leads to action potentials, the red dots, in the neurons that already have a higher level of activation because of this NMDA spike, and they produce a spike. And because of the winner-takes-all dynamics, nobody else spikes. And so the neurons that have been activated before are now spiking. This is why this is changing from blue to red here. And then, of course, because these neurons in D are spiking, the next neurons in the population B get into this mode where they have the NMDA spike. And if we now do the same thing, but we start by presenting F, then the population F is spiking, all the neurons are spiking, and the neurons in the population D that is receiving input from F gets into this excited mode, like here. But now, look, these are now different neurons. And this is why these sequences can be represented here and be distinguished, although they have this intermediate part, DB, that is indistinguishable. Now, it's just different neurons that are established, and if then D is presented, these neurons fire, and again they give this cause this NMDA spikes in population B, but again these are then different neurons. So this is how this works. And next slide, please. And now we can compare this to this much more abstract implementation that Hawkins and Ahmad did. And a typical way of comparing such learning paradigm is to look at how does the prediction error develop over the number of training periods. And in gray, you see the original model. In black, you see the spiking model. And you can appreciate that the spiking model needs more time. It needs a longer training, but then eventually learns this task perfectly. And the reason for this is probably because we have a lot more noise in the system. Everything is based on this recurrent networks that are asynchronous to regular spiking, as I have told you. And another important aspect that was also pointed out by Hawkins and Ahmad is that although we are starting from this densely connected network and we are stimulating all the neurons in the population, eventually, while training is progressing, the representation becomes more and more sparse in space and time. Few synapses are used and few spikes are emitted. And here, and this is plotted here, the relative number of active neurons in these populations as a function of the number of training episodes. And you also see here that we arrive at a very sparse representation. Again, it takes longer here than in the original model. In our paper, we do several experiments that I don't discuss here. In particular, we also look at the complexity of the sequences that we can store. These are the two that we started with, but you can also have longer sequences that are indistinguishable and also several of them. And because this learning of the sequences depends not on some total time constant or some temporal aspects of working memory, in principle, the sequences that you can store are arbitrarily long. Next slide, please. And so this is our state of the art. But the next step would, of course, be to move from this temporal model to really a hierarchical model where two things are added, and one is that we have sequences in different modalities, let's say vision and hearing, that are combined to a higher level sequence, and also that we can combine sequentially several of such sequences into higher order sequences. And this combination should have the property that one can do the processing on this higher level, but if needed, there should be a way back to resolve the details. This is sometimes called decompression, that originally are building up this structure. And this is, we know that according to Jeff Hawkins, that should work, but we are now working on a spiking implementation of this. So that's our state of the art here. Next slide, please. And in order to do this in practice, we need a simulation code. Our simulation code is NEST, that is overseen by a registered society since 2012. We are doing teaching and training on this software at different courses in international and national. Next slide, please. However, this is probably not enough if we want to advance computational neuroscience to more realistic models that combine several brain areas, as I indicated to you, or are working on higher level functions, because then the setting up the model and doing these simulations is already a task that extends the scope of a single PhD thesis. So you need to have a reliable method of describing all of this and executing this research. And therefore, we are also investing work in digitalization of the whole scientific workflow. And as you see by this circle, from extracting a model, modifying it, running it, and then feeding back changes in results into a modeling store, is implemented on a developing European ICT infrastructure for neuroscience that is called eBrains. Next slide, please. So if you want, you can already look at this site and log in and do experiments like this without installing anything on your computer. So you only need a browser, and by this have access to latest simulation technology like NEST, to models, and you can also access the compute resources. And we hope that this would accelerate research in our field and helps us to overcome this complexity barrier that what a single PhD student can do in a lab or in his lifetime is just limited, and you have problems in transferring this knowledge to the next generation of researchers to for increasing the explanatory range of such models. Next slide, please. Of course, when models are increasing, you need the respective compute resources. Therefore, we have also worked on allowing us this nest to really exploit the computers that are available. And I think this today is not a problem anymore. These computers, anyway, they are not built for neuroscience. There are other scientific fields like quantum dynamics and astronomy that require these computers, and so they are built anyway. They are available for us for use, and if you, but of course, not everybody has such a computer in the lab. But through a platform like eBrains, we can access these computers now. Next slide, please. These conventional supercomputers are not the only way of doing neuroscience simulations. You may have heard about the field of neuromorphic computing. Here, the idea is to, from the beginning, build computing hardware according to the principles of the brain, so that simulations can be carried out much faster and more energy efficient as it is possible on a conventional hardware. And here you see two completely different. Yeah, I can try to say something. Yeah, fine. Okay. So, and these two systems are really completely different. Brain scales is based on analog hardware. So here, neurons are really a small circuits consisting of capacitors and resistances. And the advantage of this system is that it can simulate networks with a speed factor of 1,000 to 10,000, whereas the Spinnaker system uses regular digital chips like the processor that you find in your mobile phone, and just has clever electronics of routing the spikes from one processor to the next. Both of these systems are connected to the eBrains platform, so you can also specify your neural network model in a restricted sense, of course, and let it be executed on one of these machines. Next slide, please. One of our efforts in making use of the Spinnaker system was to port our microcircuit model to the Spinnaker system. This was more complicated than we initially thought. It took us 4 years of doing this. Hopefully, for the next models, this is easier. However, this was really a breakthrough for neuromorphic computing, because any model that is larger than this microcircuit model is less densely connected, because this local network has the maximum connectivity that the brain has. So, and when a network is less densely connected, it should be easier to simulate this. And a few years later, the Manchester group made progress in the operating system of the Spinnaker computer, such that this microcircuit model can now also be simulated in real time. Next slide, please. And this finding that the Spinnaker system can simulate this full circuit somehow sparked a constructive race in between different groups working on neuromorphic hardware, so that there's now a stream of publications coming out reporting that the circuit was ported to a new hardware and is so much better than before, and so on. So this is now very constructive what is going on here. Next slide, please. And of course, we are also being busy and improving this on conventional hardware. So here you see the typical computer node that you probably also have in your cluster. It is one of these boxes that is 1 inch high and has 2 sockets for 2 processors. In this case, these are AMD Rome processors, which give you a total of 128 computational cores distributed over the 2 processors. Each has 64, and these are the indicated by these small boxes here. And what this figure should show you is that the situation is much more complex in terms of hardware than a few years ago. These computers typically have a so-called non-uniform memory architecture. So it is true that all the cores can access every byte in the main memory that is stored in the memory banks that you see in the background, but for different costs. So it's much more costly for a core here to access a byte here than for a core that's close to here. And also, these processors have different levels of memory that are called caches, and some of the cores share a cache, others don't, and this has a critical influence on the performance. Next slide, please. And with this system, we looked again at the performance of a microcircuit simulation, and here you see the result. This is now the real-time factor. 1 means that the simulation goes with a natural speed, and below means that the simulation is faster than biological time. And on the right axis here is the number of computational cores that are involved in the simulation. So here the rightmost dashed line is one of these full compute nodes, and the last data point here is for 2 compute nodes. And you see that already with 64 cores, real time is reached, and then we can go well into the sub real time regime. And this is of course interesting if you compare this to neuromorphic hardware. Obviously, right now the situation is again that the conventional computers are faster than the neuromorphic computers. And the second question is, what does it cost in terms of energy? You remember at the beginning of the talk we raised this question: how bad is it? And that raises the question: how do we want to measure the energy consumption? This is just one particular model in neuroscience, but we can have many different ones. And one measure that the neuromorphic community came up with is energy per synaptic event. So how much energy do we spend for every synaptic event? This is a measure that is also interesting because biologists, experimental neuroscientists can also work out what the cost of a synaptic event for a biological synapse is. And I brought you this table here that is now that is comparing different efforts of simulating the microcircuit model on different hardwares, like the Spinnaker system or GPU-based systems. And here you see the corresponding real-time factor and also the energy per synaptic event that was required. And you can see a few interesting things here. So this is 2018, and this is 2022. And you see that over these just 4 years, the real-time factor was improved by a factor of 10. and also the energy per synaptic event dropped. So we can do these simulations already much more energy efficient than just a few years ago. And also you see that presently the conventional technology is ahead. But this is, of course, it's only a snapshot. Now the neuromorphic engineers will also do their work. And what one has to consider here is that when we work with conventional computers, we have access to the latest hardware production technology. Yeah? It's just what Intel and AMD can do today. But you know that these production process is extremely expensive. Yeah? You can only afford it if you produce millions of these chips, and this the neuromorphic engineers cannot do. And so they rely on hardware technology that is always a few years ahead, maybe 10 years even, and then they have a disadvantage from the beginning. Yeah, but this thought may also mean that maybe the neuromorphic computing is a bit hopeless because consumer electronics will always move faster. We have to see how this develops. We have a question.
Why is 4 cores not double the speed of 8 cores in the last 2 lines? Is there some?
Yeah, this is because in this line here we are simulating on one node. That means formally we are doing MPI communication, but it's not really going over a wire. It's just in one node, and so technically it's just a swap of memory addresses. Whereas here we had to use 2 nodes, and that means that here the information is transmitted over a cable. Yeah, it's a super fast InfiniBand, but still you see this. Yeah, so that's a race right now where we don't know where this will end. Next slide, and this is my last one. This is just the latest photo of the lab. We are unboxing a new computer, and what is interesting about this is that this computer is now based on ARM processors, latest generation, but basically the same thing that in Spinnaker. And this has 128 cores in a single processor. The same number of cores as the machine before, but now it is only a single socket, and we are of course now very interested in whether this is now more energy efficient than the system we had before. Next slide. And so this is my summary. Cortical structures are conserved over evolution and modalities. There's a dual universality that raises hopes for generic principles. We now have cortical models that we can use as building blocks and platforms for further studies. I argued for the need for brain scale models and the need to formulate brain algorithms fast in space and time to overcome this energy problem. And I discussed sequence learning as an example based on the TM model. I showed you that supercomputers are ready for use by us, but also fully digital workflows that may be required to overcome complexity barriers. I showed you that the simulation of a microcircuit was a breakthrough for neuromorphic computing, and that achieving sub-real-time performance is possible for networks of natural density, and the potential for reducing energy consumption of computing. Thank you very much.
Thank you, Martin. I guess, is there any question here while you take there's questions on the chat? But anyways, first here, anybody has a question? There's someone in the chat. Anybody? Wait. I'm sorry. Okay, so yeah, because there's 2 here, but sorry. Okay, whatever. Go with Felipe. It's fine. Felipe? Tenían la mano levantada.
No, yo no- estaba aplaudiendo, disculpe.
Sorry, all right. Pedro, please.
I have a question about the sequence learning. I understood that basically the different sequences will rely on a sequence of firing of different parts of the network. So what happens if you, what do you think may happen if you just use one sequence of, or don't use the other sequence, will forget the other sequence, or how robust is the memory of the sequence they have done?
Yeah, so the learning that we used here is structural plasticity. Yeah, so we assumed that every neuron can touch every other one in the local microcircuit, and that by structural plasticity a synapse is formed. But maybe you know better than me. I think this turnover of synapses is much faster than we thought about a few years ago. So I think over a time course of a day, this then may change. So if this is not used, not replayed, not sustained in any way, then the synapse can go away.
But it won't be a catastrophic.
No, not immediately, but it would fade away.
Okay. Yeah, but at least it will stay. Okay.
Yeah.
Is the capacity boundary, how many sequences can you be in?
Yeah,
There's the limit?
there's certainly one, but we have not tested this. Yeah, and also the real simulations that I showed you were not on the microcircuit level, but with much fewer synapses and neurons.
So let me jump because I had a related question. So when you were presenting the model, I was like, how in the hell does this not interfere between the sequences? And then I understood, obviously, you're using subspaces of the network for different sequences.
Yeah.
But that probably really depends on your sparsity parameter, right? So I was wondering whether if you toyed around with the sparsity parameter to see how interference occurs in that sense. I guess if you augment the probability of then you will have a much smaller space for actually another sequence to be represented, right?
Yeah, certainly there is a limit. I just brought you these few examples to convince you that it's cannot only learn two.
No, that's actually super cool. Yeah.
But where the limit is, we don't know yet.
And then the other question that I had related to that is, so this is what we were discussing before your talk. We know that we learn compositionally, right? So in the two examples that you showed, there was like whatever, I don't remember exactly the letters, but let's say for instance ADB and ADC, right? Or EDC. And so the D's, they're the same actions or the same order sequence. Yeah. But right now, you almost considering them as being like different neurons, although they're tuned to the same stuff. You know where I'm going with this, right?
Yeah.
And so I was super interested by the hierarchical part of it, right? Because hierarchically you can extract that information, right? And then reconstrain it afterwards.
Yeah.
Is that the idea with the?
Yes. That's why we need this hierarchical aspect to make full sense of this. And that would then also have aspects of the compositionality that you mentioned, that we can then have an auditory sequence bound to a vision sequence, right? And let them represent the same object, and then maybe you can work out completion, like you hear something auditory, but you can then imagine how this in vision looks like. Yeah? You hear an ambulance, and you get the image of an ambulance deck.
Yeah, that's super cool. It's nice.
I think you already. But so I also have another question. But so you talk about three different modes of the network of the neurons to accomplish this. But maybe can you expand a little bit there, because I couldn't get it. Why it's important? How you justify it?
Yeah. Can you go maybe back? Show for your slides. So this is the distinction between the original Hawkins and Ahmad model. They require that the neurons have 3 states. One more here. So the Hawkins and Ahmad model require that the neurons have 3 states. There's quiescent, predictive, and active. Yeah. But in our model, we just assume the regular integrated fire dynamics. So either the membrane potential is subthreshold, or you have a spike. Yeah, and then you have a reset and it goes on. This predictive state that Hawkins required is completely implemented in our case by this NMDA spike in the distal basal dendrite. Yeah? So that here A is presented, therefore A is predicting to the neurons in D, and it is given an input to the distal basal dendrite that is large enough to cause this NMDA spike. So the neuron becomes more excited, but it still cannot fire a spike. Yeah, but this is long lasting. Yeah, this has a time constant of 100 milliseconds, and then D is presented to the system, and it hits on this already more excited neurons, and then the spike is fired.
Then, so these neurons have 2 compartments?
Yes, if you want to consider it this way, yes. Okay. Yeah.
Yes. And it is the winner takes all because it was driving on top.
So the winner takes all comes from the fact that all this is a recurrent network of excitatory and inhibitory, and the inhibitory neurons, they receive input from all the excitatory ones. So once enough of them, of the excitatory ones, fire a spike, then the inhibition kill comes in and kills all the others.
If you invent a new letter, right, then is there a simple way that you can, I mean, somehow the structure of this model is based on the structure of your alphabet, right?
Yeah, yeah.
So
yeah, this is something that we pre-assumed. Yeah, we assumed that there are these 6 receptive fields. Yeah, that we have 6 symbols, and yeah, that would then be an additional learning step if you want to form new ones.
So that will be like a community formation, like they will gather because of similar signals in a way. You assume that they will form some equal probability.
They're basically tuned to that abstract signal.
Yeah.
Yeah. I had maybe other questions. I don't know. So the other question I had was, so you're implementing STP, so that also is strongly related to the timing of the sequence, right? Like the input itself. Meaning that depending on your NMDA parameter, like the decay parameter, at some point you can input a second stimulus, and you won't create the link.
Yeah, exactly. Right. Yeah. This is the weakest point about this model, that this only works for basically for sequences with a fixed speed or the maximum distance, and it's left unexplained how we can work with so many sequences of different lengths.
Actually, so you probably know the work of Cohn and Chauvin, and they actually also have that kind of decay parameter to link these two say population of neurons, right? But they also have that problem with like basically all depends on the decay parameter itself.
Yeah, and that's not something I would like.
But it's I guess one of the constraints of STP on its own. Yeah, no, that's interesting. Then maybe if nobody has another question, there's this other thing that I think it's actually quite fascinating, because say I'm in context light blue instead of dark blue, right? And I start A, I know that my last node will be E, or my last stuff is E, right? And in the other one, it's C. And there's empirical data that shows that you can slightly decode that in time, right? Because I know that I'm going to produce that.
Yeah.
But here it's really close to like A is going to predict D, but it's not going to predict B in time, right? And so I was wondering whether there's parameters in the network that allow to kind of control the flow of the attractor networks, like maybe dilute it, in that sense.
Yeah. I guess if you set the parameters of structural plasticity right, you would automatically get this after a while. Yeah? Because every neuron is touching every other one.
Yeah.
So why not, if A and E occur in a correlated fashion, you would eventually create shortcuts.
Yeah.
Interesting.
So it's just last question. Is there a theoretical? I guess this is a repetition of another question. A theoretical bound of the capacity of the network, something like with an upper bound, like
certainly there is an upper bound. Yeah, once you have used up all the synapses here in your sequences, then
combinatorial.
There's within the Hopefield network framework, you have a way of actually knowing what's the limit of the. I don't know here with like the spiking framework, but in the Hopefield network you do have that.
So there are works on it. Whether this can be transferred to this situation, we don't know, but it's
because you can the limit of memory, so you have more capacity. Yeah, so memory should improve capacity.
Okay. No more questions on 9 here.
computers.